Hello, my name is Abdu Hijazi and I will be showing you this video about the role of GTP in the formation of the translation initiation complex in bacteria. This video is made for MCDB 427, also known as Molecular Biology at the University of Michigan, and it is based on Figure 17.8 from Robert Weaver's 5th edition of Molecular Biology. The initiation of translation in bacteria involves several steps, but we will be concerned about this part only for the figure. It's at the top, and it involves the 30S initiation complex. The 30S initiation complex is made of the 30S subunit, 3 initiation factors IF1, 2, and 3, GTP, mRNA, and aminoacyl tRNA. Now the aminoacylated tRNA, shown right here, is called fMet-tRNA fMet, and it is a special tRNA. We call fMet-tRNA fMet since it is an initiator N-formal methionine tRNA charged with N-formal methionine amino acid. So the IF3 comes in first to prevent the subunit reassociation, followed by the binding of IF1 and IF2. Other data showed that IF3 was the main factor that mediates the binding of mRNA to the 30S subunit, while IF2 was the main factor that mediated the binding of fMet-tRNA fMet to the 30S. Now you notice that right here, IFP is bound to GTP, or IF2 is bound to GTP. The GTP is required for IF2 binding to the initiation complex, and we'll show that later. So Faconding and Hershey wanted to know if the hydrolysis of GTP actually had any effect on the binding of IF2 and fMet-tRNA fMet to the 30S. So they performed an experiment where they mixed tritium labeled fMet-tRNA fMet and 32P labeled IF2, which got their label from phosphorylation by 32P labeled ATP, with 30S subunits and AUG, which was used as an mRNA substitute. Then they added either GTP or another non-hydrolyzable analog called GDP-CP, which has a methylene group instead of an oxygen between the beta and gamma phosphates. So you can see right here, this is the non-hydrolyzable analog. This is the beta phosphate, and this is the gamma phosphate right here. Usually you have an oxygen in GTP right here, but a methylene was placed here, which makes it non-hydrolyzable. And then you separate the initiation complexes by sucrose gradient ultracentrifugation. Now, before we get into the experiment and results, I want to briefly talk about the sucrose gradient centrifugation technique. This technique is commonly used to isolate and purify biomolecules and cell structures. In this example, the sample used was ribosomes. You can see that the sucrose gradient is increasing from the top to bottom. Then you add your sample at the top of the gradient, right here, and then you centrifugate at very high speeds. And here, when you get your different parts, you collect them in different fractions. And in each fraction, you're looking for something, like it depends on your experiment. And for the experiment in the figure, their lysate that they put on the top, or their sample, was initiation complexes. And what they were looking for in each fraction was the radioactivity that represented both the IF2 and the fMet-tRNA fMet. Now going to the figure, the graph in blue is quantifying the amount of IF2, and the graph in red is quantifying the amount of fMet-tRNA fMet. Now looking at the left figure right here, we can confirm that fMet-tRNA fMet and IF2 actually do bind the 30S subunit. You can see right here that the 30S is located at this fraction right here, and Both graphs show radioactivity in the fraction containing 30S, so right here and right here. So this brings us to our first conclusion that IF2 and fMet-tRNA fMet bind the 30S subunit. And comparing the left figure where GTP that can be hydrolyzed was used right here to the right figure where non-hydrolyzable GTP was added, very similar results were obtained. Well, you can argue that the binding of IF2 was a little bit better in the non-hydrolyzable analog one, but since they were very similar, we can conclude that the hydrolysis of GTP was not important for the binding of IF2 and fMet-tRNA fMet to the 30S, because you can see that there was no change in results or like a slight change. Well, they're still binding when non-hydrolyzable GTP was used instead. You do not have to worry about these parts right here at the end. These are just showing free unbound tRNA, well, which they show at the top of the gradient. And there is one experiment not shown here that I would like to talk about. And it is like, what if no GTP was added? You would see graphs looking like this for both the IF2 and the fMet-tRNA. They would just appear at the top of the gradient. Which brings us to our third conclusion that GTP is required for the binding of IF2 and fMet-tRNA fMet to the initiation complex. Now, if you're wondering what GTP hydrolysis actually does here, later experiments showed that it plays a role in the removal of IF2 from the ribosome, but I will not talk about those experiments. Thank you for watching this video, and always remember to go blue.